And I was just not very thrilled to be in a lab slicing mouse brains. A honeybee colony, it's considered a superorganism. Together, the whole superorganism has these emerging properties of the superorganism that are higher level than what they would be for the individuals in the colony.
Hi, my name is Irfan Vafai with Texas A&M AgriLife Extension. Welcome to Talking Bugs, a podcast about entomologists and their research. In this episode, I chat with Dr. Juliana Rangel, an associate professor at Texas A&M University. She runs the Honeybee Lab at Texas A&M University, which can be found at honeybeelab.tamu.edu. They hold annual trainings and provide resources to help beekeepers. Dr. Rangel has published over 69 research items with 691 citations and 11,247 reads of her works, according to ResearchGate. Her research is mostly related to pollinator and bee health. I hope you enjoy. Thank you so much for joining me today.
Thank you for the invitation.
Absolutely. So I can see you are obviously working from home as well. So how's this whole thing? You were mentioning a little bit earlier that I guess you're kind of used to working from home now for a little while.
Yeah, so I was telling you earlier that I have a 7-month-old baby. So in the fall, toward the end, I was on maternity leave. And then this spring, I was on sabbatical leave, or what they call here faculty development leave. And so we were gone for the month of February, and part of early March, we were in Australia visiting
Nice.
a collaborator when this whole pandemic thing was kind of exploding in China mostly. And
Ooh.
very little was heard in the news about any other places, maybe one or two countries in Europe. Italy was kind of getting started with their big issues, but so it was surreal to be there and go through that part of the pandemic when we were so far away. And
You were in Australia
luckily,
in around February then, or when was
all
that?
of February
Oh
and
my
part of
gosh.
March.
Oh
Yeah.
wow, yeah.
And then we got back before they put all the travel restrictions.
Yeah.
So we
Oh
just
my
made it
gosh.
back in time, and we quarantined ourselves. And
So I think that would have been
so,
just before Tom Hanks was quarantined, I think, in Australia for
yes,
14
that's
days.
right. It
Yeah.
was exactly, yeah, just before.
Just missed him. Just missed him.
That missed them, yeah. Um,
Neat.
so it's been now over 6 months of working from home, and it's been interesting. Of course, I have to juggle the family life, the home life. Luckily, I have a very supportive spouse, but he can only, take the
Do so
kids
much.
and do so
Yeah.
much. So I've actually, all things considered, I've enjoyed my time at home because I get to interact with my family more than I used to. So there's trade-offs, and we now can conduct some research at our lab. And so I've been going to our field laboratory a couple of days a week. So it's kind of breaking the quarantine
The monotony of
monotony of being home, stuck at
yeah.
home all the time.
Yeah,
So
exactly.
yeah.
And you started by doing your Bachelor's of Science in University of California, San Diego, in Ecology,
Mhm.
Behavior, and Evolution. Did you already have a specific career goal in mind when you were doing the undergraduate degree?
Not precisely when I started. I migrated to the U.S. Right after high school. I grew up in Colombia, South America, and um, I had actually, I lied. I did a couple of semesters at the university after graduation in Colombia, but I couldn't really afford to go to another city or state. And so the only thing that they had close to biology at the local university was environmental sciences. And so
Okay.
that was my major there. And I think I would have been fine with that, but I was mostly interested in biological systems and studying specific organisms and not necessarily um, broader environmental impact kind of classes. And so when I moved to the US, I had this open opportunity to study pretty much anything I wanted to. I started by going to community college, though. So I did 2 years of just science prerequisites. I knew I wanted to do biology for sure. And as soon as I transferred to the University of California, San Diego, they had several biology majors or types of degrees, and one of them was ecology, behavior, and evolution, and that's the one that was the most interesting to me. I was working in a lab, as an hourly research assistant in a neurobiology lab that was working on trying to understand the neural mapping in the brain of mice. And so I was working,
Ah.
helping with studies looking at the brain and how it works. And I was just not very thrilled to be in a lab slicing uh, mouse brains. And I
Just not
just
your
did
cup of
not,
tea.
I did not see that, like neurobiology was not necessarily what I was interested in doing. I knew I wanted to do something um, outdoors, more kind of at the whole organism level,
Yeah. So that's very interesting
population level.
because that's when you transition from your undergraduate degree into a PhD in neurology and behavior, but you're not
Yeah,
studying it on mice. You're looking
right.
at, hive behavior and social insects.
That's right. So when I was an undergrad, on top of doing this research with the mice in the neurobiology lab, I was working for research credit at the B lab there. And
Oh,
that
okay.
professor had just started that semester that I was his first ever student. He's now a very popular and accomplished professor, but I was his first ever
Wow.
undergraduate
Who
assistant. And
was that?
James Nye,
Okay.
and he studied at Cornell University in neurobiology and
Ah.
behavior. So he, when it was time for me to apply for grad schools, he suggested that one, and I applied to a few other programs, and that's the one that I chose to go to because um, I would be able to work on similar questions that I was asking as an undergraduate research assistant with stingless honeybees from
Oh,
Brazil. So
okay.
that's how I got started working with bees, was helping Dr. Nye with his research on stingless honeybees.
And so then
And
you go under the supervision of Dr. Seeley. Is that right for your
yeah,
PhD at
that's
Cornell
right.
University? And he studies swarm intelligence. So I
Hmm.
thought this concept was really neat. Can you tell me a little bit about swarm intelligence?
Yeah, so if you think of a honeybee colony, it's considered a superorganism because each little bee cannot really survive on her own for more than a few days without being part of the colony itself. And every bee, based on her age, has a specific task. And so combined together, the whole superorganism has these emerging properties of the superorganism that are higher level than what they would be for the individuals in the colony. Just like you can think of um, uh, the cells in your body coming together with emerging properties to create organs, and those organs coming together to create the emerging properties of the of the human or the whole body on of the whatever animal you're referring to. And so honeybees reproduce by swarming or splitting into two units every spring. Right now, it's the prime time for colony level reproduction, and so they swarm. And
Uh-huh.
these swarms are um, comprised of thousands of individuals from the colony. Somehow, even though it looks very chaotic when the bees are undertaking this swarming behavior, looks like it's just a cloud of bees, they have this emerging property of combined intelligence that tells them exactly how they should operate to first cluster in the tree branch, which is what most people find,
See, and
see in
or in
their,
the case of an email I got today on their water meter.
or water meter,
Yeah.
or inside cavities in their home. But typically, that whole process of swarm intelligence is just the emerging properties um, of the organisms within the colony coming together to create this um, uh, the
This swarm
superorganism that
intelligence,
the
essentially.
swarm, yeah, the big swarm that almost looks like a big brain with all these um, neurons thinking and firing and making decisions, and so it's a decision making machine.
It's
It's what a
amazing.
colony is.
And it's amazing seeing how they're trying to, how understanding that can have such implications in technology. Looking at, there's like these drones that you see up in the air that they make certain image formations in the air, and they're using swarm intelligence. They all
That's right.
communicate to each other. If there's wind that affects one, they all need to communicate to respond, respond, correspondingly. So it's very neat to think of, it's a bunch of bees, and to a lot of
Right.
people, it just looks like a chaotic movement of bees, but it has all these crazy and very intricate implications into how they make decisions and how they move like one mind, like you said.
That's right. And if you look closely, which most people don't really have the time to do, but if you're a graduate student doing a PhD, that's kind of what you're stuck doing. Then, if you have time and look closely, you pay attention to all the um, behaviors that they're performing, the signals that they're producing, multimodalities that they're using, olfaction, um, vibration, vision, vibration, acoustic kind of hearing or detection of sound or vibrations, um, and all combined allow for the coming to decisions that are kind of life or death situations for swarms, for example, of when to move to a new home and where to go together as a unit. They never really have a split decision where a swarm will split into one group will go to one place and the other one says, "Oh no, we don't like that one. We're just gonna go to the other place we liked." And they never really do that. They all come to an agreement um, into where they're gonna end up going as their new home. And so a lot of computer scientists and engineers use honeybee decision making, especially during house hunting and swarming, as a model to understand and see how they could improve the
Hmm.
algorithms that they use from the intelligence of these animal um, system swarms to, as you said, use them in computational analyses and um, for drone technology and for all kinds of other emerging technologies. So,
That's pretty amazing. So what actually inspired you to get into this in the first place?
well, I was working with honeybees and Tom Seeley, my PhD advisor, had been working on um, Appledore Island, which is a small island in a small archipelago called the Isles of Shoals, off the coast of New Hampshire and Maine. And he's been working for 30 or 40, now 40 years in these questions about how honeybee swarms decide where to move to found a new nest. It's not a trivial decision. It's actually a very important decision because only 1 in 5 swarms makes it over winter. It survives its first winter.
Wow.
And so selecting a good quality home um, can be a matter of life or
Life and death.
death for these swarms. So he had already explored, and he continues to, I think, to explore questions regarding the house hunting of once the swarm is on the branch, on the tree branch that you find, this time of year. Then he looks at what's going on in that swarm cluster, and what behaviors the bees are performing to come to that whole decision-making process of where to move. So but the question remained of whether or how the bees were selecting to leave their nest, leave the cavity that they were occupying, to go to that swarm cluster place. So the first part of the equation, which would be to leave their house to embark on the new journey to their new home, because they typically 95% of the time they never come back
Hmm.
to their old home. They just go with the mom, the old queen.
Yeah.
She's the one that leads the about 3/4 of the workers in a colony when colony swarming, at least when they're issuing the first swarm.
Yeah.
Um, and um, and they just go with her. They go with the queen, and they cluster in the tree branch. Then they do these other things that Dr. Seeley had found, and then they finally go to their new home. So I was able to provide the missing link of what happens before they leave the house and or the hive, and how they do that to
And that
go
was
and cluster.
that was the
That was
paper
my
from your,
whole that was my PhD dissertation. We published 6 papers out of that looked at the behaviors, the significance of what we call the swarm fraction. So the colony, why does it split more or less in like 1/4 of the workers stay and 3/4 leave? Why not 50-50 or
yeah.
30-70 instead of 70-30? And there are evolutionary optimal um, numbers,
Ratios or yeah.
ratios that benefit both the colonies, that the bees that stay behind and the ones that leave. That break of about roughly 1/4 staying and 3/4 leaving, we showed with a mathematical model that it's the ratio at which the fitness, the reproductive fitness of both units of a colony that split increases their chances of survival to
Wow.
the next year.
That's pretty amazing. And so it
Yeah,
seems like you had an opportunity in your undergraduate that kind of inspired you in this field, and you just kind of follow those opportunities, and has led you kind of where you are now. Would you say? I mean, it's
that's right. Yeah, it's
yeah,
all been kind of, I don't know, serendipity. Things, just opportunities have kind of appeared, and like the saying goes, you just mm, prepare yourself for those opportunities and seize
yeah,
them. And I've just been incredibly lucky
yeah.
that I've been
It's
able to do all of this stuff.
I feel like I should just call this podcast "Serendipitous Entomologists" because everyone I've spoken to, it's kind of that's how they found the path. You know, it's
Yeah,
in their undergraduate degree, they found something they were passionate about, and either it was the insects themselves or it was a certain concept like neurobiology, and
that's
insects were
right.
the system, and
For
it kind
me,
of
it's
all
the
connected.
latter. So it was not like I was a bee lover all my life and I wanted to study bees. I know a few people like that, but for me, it was more the type of questions that were being asked, and I just then fell in love with the system, with the subjects, the honeybees.
Yeah, and so
Yeah.
now you run the honeybee lab at Texas A&M, right?
Hmm.
And y'all do some pretty awesome research for honeybees, whether it be looking at agrochemicals in the wax of beehives, or looking at the types of pollen that honeybees collect and how important those might be, to a paper we're going to look at here soon, which is potential vectoring of viruses by other species in and out of
Hmm.
hives. And what are some other services and or programs that the Honeybee Lab provides?
Sure. So when people ask what we do, because if you read the types of papers that we publish, there's a wide variety of topics, and I think what links them together is that I'm interested in understanding the factors that affect the health of honeybees,
Mhm.
both wild and managed honeybee populations. So I'm not really hugely involved with native pollinators at this point, because it is an apiculture position, and so I've tangentially worked with um, some pollinators, non honeybee pollinator projects, but ours focus mostly on honeybees, and so um, we typically have a staff of maybe 11, 12 people at any given time, including 5 PhD students typically.
Wow.
And we do mostly research. I mean, my position actually doesn't have any extension component to it, which people are surprised about, because I do a lot of extension related activities
Hmm.
for the public, because there is such a strong need
Yeah.
and desire for those programs. And unfortunately, we don't have an apiculture extension um, person at the Texas A&M system. So I fill a lot of those voids myself. And then so it's mostly research, and I teach 2 undergraduate courses in honeybee biology and beekeeping, but those are of course for enrolled students. So um, I give maybe, I would say, 10 presentations per year to local, state, and national beekeeper organizations, and actually
Oh, nice.
international nowadays. Um, and we offer, well, this year was we had to cancel everything just like almost everyone else, but uh, we offer a um, queen rearing workshop every May or June, and it's very well attended. We thought that maybe the first big enrollment class 5 years ago was going to be it, because um, uh, so one of the main lines of the research that we do that I'm most interested in is the reproductive biology of queens and drones.
Okay.
And so
And
part of
drones being the
that,
male
drones, the
bees.
male honeybees, yes.
Hmm.
And so um, one thing that you can, as a beekeeper, you can do is produce your own queens. There is kind of an inexpensive uh, way of producing queens, and so we have a queen rearing workshop. But when I first um, talked about wanting to do one of those here in Texas, people thought maybe there wouldn't be interest because of the fact that we have the Africanized honeybee here in
Okay.
Texas, that people would not be interested in rearing queens that would potentially be of Africanized stock. And um, the truth is, people are well aware of that fact, and still they want to produce locally adapted queens. And they're interested in the biology of queens, and the art of queen rearing, which is the name um, of our workshop, incidentally. And so there's just always going to be, seems like, interest in producing locally adapted queens. So that's our kind of most unique program that we offer. It has about 50 people enrolled, and we typically bring a couple of guest speakers and presenters. So it's an all-day event where we do hands-on for half of the day and then lectures the other half of the day.
Nice.
But we're also, in the last 3 years, we are co-organizers of the fall Brazos Valley Beekeeping School.
Okay.
That is put together by our lab and the Brazos Valley Beekeepers Association.
And that one fills up pretty quick too.
Yeah, so we used to just be guest instructors at the event when it started about 5 or 6 years ago, and they outgrew their first space, so then they moved to the Agri-Life Center when we started organizing it on the main campus of A&M, and apparently this year they've outgrown that space, or
Wow.
we, I guess, and so now we're going to have to do it in a different location that has more occupancy allowance
So
for
that's
the
that's a few hundred.
classrooms.
That's a few hundred
Yeah,
people.
that's including volunteers and instructors. It's about 500
Wow.
people or so. We also teach at like the usual gigs that I am very happy to help with include a bee school that's put together by the Austin Area Beekeepers Association in January, and that's again like an all-day bee school. For most of these have a beginner and an intermediate track for beginner and intermediate beekeepers. There's also a few advanced commercial beekeeper kind of presentations, but it's mostly beginners and intermediate. There's one that was canceled this year, probably for the first time in, I think they've done it for 12 consecutive years. It's the Central Texas Beekeepers Association Bee School, typically in March or April. It's out of Brenham.
Okay.
And they can, in their highest enrollment years, they can have about as many as 1,000 people.
Wow.
That day.
So how
taking
can
classes
people
about
find
bees.
out about some of these programs if they're from those areas?
Everything. So, well, selfish, pitch would be go to our Facebook page, Honeybee Lab. You just look up TAMU, TAMU, Honeybee Lab, because we post everything on that site, everything that is coming up in the region, nationally, internationally, all the papers we publish, or we collaborate on, all the awards that are won by our students, because they're just tremendous, very successful in earning awards and publishing. And another one is which just got canceled, and Tom Seeley was going to be the speaker, so hopefully he can be back next year, is the Texas Beekeepers Association Summer Clinic. Kind of same type of deal, but it's in June, and then the Texas Beekeepers Association Annual Convention, which is in November. And so that's the umbrella organization for the state, and they have a few more commercial beekeepers involved in the leadership roles and the management of the organization. And so we're kind of spread. It's a way to spread. We can't really go that far because Texas is huge. We can't really go to every county beekeeper club presentation, but we can go to the regional ones and hopefully reach more people that way.
I think that makes sense. Texas is a very big state.
Yeah.
So certainly reaching all of it is a little impractical.
Yeah.
So as the person who kind of runs the Honeybee Lab, I've got to ask you what you think of these like murder hornets that have made the news.
Well, that's an infamous common name. Just like it was, they called the Africanized bee the killer bee. It's doing a disservice to to the field of entomology and to the conservation of these species. But the Asian giant hornet, also known as the murder hornet, has given us an opportunity to revive the conversations about the importance of pollinators and honeybees. And so the Texas A&M College of Agricultural and Life Sciences immediately responded to people's questions about the Asian giant hornet by creating a task force. I don't know if you heard the news, and there was a big press release about the task force, and where we have constant discussions about how to handle the potential invasion of this Asian hornet if it ever came into Texas, and what would happen. So the name murder hornet comes from the fact that they, just like other wasps and hornets and vespids, they eat other insects. They're carnivorous, so they murder other insects. And in the case of the Asian honeybee, they can truly go and invade a colony, and in a matter of a couple of hours, if the colony is not well defended and there's too many hornets to deal with, they can decapitate and kill a colony in a matter of hours, and then they go on to eating the tasty larvae and pupae and bringing them to their young. So hence the name
murder hornet.
But the thing is, in Asia, where they're from, they invade colonies of the Asian honeybee, Apis cerana, which is a cousin of our Western honeybee, Apis mellifera. So Apis cerana has co-evolved with the giant hornet and has evolved ways to combat their attack. I don't know if you've seen the videos of the Asian honeybee balling the hornets and basically cooking them through social fever that goes, they vibrate their muscles, and they were able to generate so much heat that they basically cook the hornet inside the bowl, but the temperature is just low enough that they don't cook themselves.
Right.
So that's a brilliant strategy for combating this invader. The Western honeybee does not have any issues in its normal range in Europe and some parts of Asia, Western Asia, etc. They don't have hornets to deal with like that, so they haven't evolved any mechanisms or strategies to combat them. That's the problem, that when they invade areas where Apis mellifera lives, they can go in and wreak havoc in those colonies, and the bees don't really have a whole lot of options.
I guess, if you ever have that opportunity, if they do establish, and that's a big if, and we're able to get some of those Asian honeybees, that'd be a really interesting swarm intelligence difference to study kind of between the Asian honeybee and the European honeybee or the Western honeybee.
That's right. And very few people have studied swarming behavior in other Apis bees, because just like with almost everything biology related, most of the biodiversity in the world exists in tropical and subtropical regions, but most of the resources are in areas of higher latitudes where there's not necessarily as much biodiversity. So there's not a lot of university support for people studying all those really cool Asian bee species that are similar but not the same as the Western honeybees that we look at. So
as a research scientist, a graduate student mentor, what are some of the skills or life values that you think have been most important in your career?
I would say seizing those opportunities that come every so often, and being prepared professionally, educationally, and emotionally to take them. I think is um, quite important, not being afraid of failure or of what might happen in the future. For example, starting out a lab and having only limited resources, some people could have a strategy of being very minimalistic or frugal in the way they invest their little resources because they're afraid of what happens in the future. If I don't have it, run out of funds, then I won't have the ability to fund any more research. But my approach was, well, if I make a good investment now, the first or 1 or 2 years, hopefully, you need a little bit of luck, but you also have to have a body of work that can try to help you get more funding from federal and state agencies, which has been the case for my lab. So seizing the opportunities and not being afraid of the unknowns that come in the future is one thing that I would bring as a piece of advice. Surrounding yourself with people that can um, provide positive feedback is important. The unofficial mentors that you
Mm.
can rely on, because if you know it, and most people have probably gone through something similar, that graduate school is not a sprint but a marathon, and you
Right.
um, have to go through a lot of ups and downs. And um, I don't shouldn't say this out loud, but I don't take my job so seriously that uh, that I that I only live for my work. I have a life outside of the lab and the office, and I think I
Hmm.
instill that in my students, that they have to work and be very productive, but also they have a personal life and they have a life outside of the lab, and they don't have to be at the lab 12 hours a day to be productive. Um, One that I wish I was better at is time management.
Hmm.
Uh, I can only advise people that if they can find ways to manage their time better, that's a skill for life. I struggle with it, and a lot of people do too. Um, so it's for me a learning process, and it will continue to be. But that would be one piece of advice, is trying to find strategies for managing time. Um, in the case of graduate school, I would suggest, which I did when I was a graduate student, is two things: to take training in grant writing. Um,
Did you teach
and
that course?
I teach a graduate course that's it's quite popular,
Hmm.
because it shows how people the whole art of writing grants proposals, which is an art, and you always get better at it with time. You have to learn how to take the failure of rejection, because most likely your grant proposals are not going to be funded, especially not the first time you submit them.
Yeah.
So taking some sort of, if there's now official class, at least go online and look for or read a couple of books on grant writing, um, and either create or join a writing club. I'm part of one for women in agriculture, and we meet once a week. Uh, and it's a way to build relationships with people outside of the department, with other female faculty, but also it kind of makes you write. And I would say that I would suggest that students write, little by little as they go, as their projects evolve. If they finish the methodology part of an experiment, I encourage my students to write that up and not wait until the last minute to write the whole. not to not think that they have to do the whole experiment and be ready now to write the whole manuscript in a matter of a month or two under a lot of pressure. Instead, you could just write a little bit at a time as things progress. If you already know what methods you're going to use, if you think that you know what body of work inspired you to look into those questions so that you can start writing parts of the introduction or the discussion. I think that's another piece of advice. And to seek help when needed, to not be afraid to ask for help, not be afraid to say, I screwed up, or I don't know how to do this, or I did it wrong. Because it's also it's a learning process. And the sooner you can ask for help, I mean, I encourage people to work independently, but also to recognize or realize, hopefully early enough, when they're not getting the answer they need, so that it avoids wasting time and resources unnecessarily.
Right. And I think going back to even just writing, I think that's one of the things that I've been working on a lot, is trying to make that time to write, whether it be grant writing or manuscript writing, because it does take quite a bit of mental energy. You have to spend a lot of time reading. And once you finally read a whole lot, then you can maybe start formulating some sentences and some paragraphs. And then you step away from it for a week. And then if you want to write again, you have to go dig back into that literature again and get your head back into that space. And so for that reason, it can feel non-productive, because, you'll spend a lot of time and write a paragraph, whereas I can spend a couple hours and plant, a couple hundred plants in my greenhouse, So I
I
think for that
know.
reason, in terms of time management, that is one of those challenging ones. And I think it's nice to hear that encouragement of, that's one of the things that we all kind of need to work on.
That's right. I I see a lot of people that have writing anxiety, and
Mm-hmm.
that's very valid. You have to just work through those anxieties just so that you can um, find triggers of that mental block, try to overcome them so that little by little, you can get moving. And the more you write, the less anxious you become.
Hmm.
Even if it's just writing, reading papers and summarizing them in your own words, things like that will help you create the introduction later on. So another piece of advice would be to write anything and everything you need, and then to um, edit later,
Hmm. Yeah.
so that,
Uh,
especially if you have a lot of that anxiety, so that you can see, at least, you can just visually, you prepare yourself, you see some wording on the piece of paper, and that's already giving you a little bit of encouragement. If you keep deleting everything you write because you don't like the way it looks, then you're gonna end with a blank page and a lot of frustration.
Yeah, I've
So
heard that strategy before, is just write down what's on your mind
mhm.
and just keep writing, and then go back and edit if needed, or you can restructure from there. But at least you get that on the paper, so it's a lot easier
Right.
to get a good start. And so that brings
Hmm.
us, I guess, into a somewhat of a recent, well, actually very recent, 2020 publication that is Alexandra Payne et al. So
Hmm.
you are the senior author on there, and it's
Hmm.
entitled "The Detection of Honeybee Associated Viruses in Ants." And
Hmm.
so in this study, you're looking at potential intraspecific virus transmissions. The idea that you can have viruses that go between different species, which
Hmm.
in our case of COVID-19 can be quite relevant and interesting as well.
That's right. Yeah.
And so ants, I guess, are a very common pest in apiaries. And
Hmm.
so the purpose of this was to document these interactions between the ants and the honeybees, and
Hmm.
then also to see if any of the 6 commonly occurring honeybee associated virus are present in the ants that you collected near or far from these apiaries. So
Hmm.
can you tell me a little bit about what are some of these bee viruses?
Yeah, sure. So, honeybees have dozens of viruses attacking them, and the better we get at um, with our molecular techniques to find these viruses, the more we find, but it's not that they're getting new ones, it's that we're getting better at detecting them. So one of the most notorious one is called deformed wing virus, and it's more notorious because um, well, it's present in most colonies of the Western honeybee, and it is one of the few that is symptomatic. So, when there's high levels of the virus in a colony or within a bee, then, as the name indicates, it deforms the wing of the bees during development, and when they emerge as adults, their wings are crippled and they're kind of um, useless. Basically, they can't straighten out their wings, and so they can't fly, and they die a few days later. So they are not productive. They can't really do much within the colony. But there's other important honeybee virus, chronic bee paralysis virus, Israeli acute bee paralysis virus, sac brood virus, and they occur um, eh, in potentially fewer levels. Um, and so not all colonies have all of them at once. But the honeybee, the biggest problem we have still in apiculture today is the varroa mite. It's a parasitic
Mm.
mite that attacks the uh, the bees, lives within the honeybee colony. They can't really live outside of the honeybee colony, um, and they feed on the developing bees and the adults. They feed on the fat bodies of the bees. And while they do that, because they have sucking, piercing mouth parts, while they do that, they insert and transmit a lot of those viruses. And so they, the number or the level of the viruses is much higher when colonies have high varroa levels than when they have low varroa or no varroa at all. And so um, it has been shown that colonies that collapse during the winter, they typically tend to have higher levels of viruses and more viruses, combinations of viruses than those that don't that survive. So it's a big problem having these viruses in
And so,
colonies.
if some of the bees have the virus, why don't they all just interact with each other to get herd immunity? Why don't they all just go out and socialize and rub on each other? Does that work? Can they get immune? Can they adapt to these certain viruses?
So they can, to a certain extent, there's some viruses that are less virulent than others, so they kind of evolved to have lower virulence, so they don't make the bees as sick, and therefore the bees can still have the infection, but still work relatively normally and have the ability to transmit the virus to
Transmit.
other bees. So yeah.
Wow.
Um, but what's interesting that we've been looking into, it's my student Alex Payne. She is um, I have to give her a shout out because this is her project. She
Hmm.
envisioned it, and she received a National Science Foundation fellowship to do
Wow.
this work. Um, we've learned in doing our literature search that there's more of these viruses, or the same viruses occur not just in honeybees but in other, well, first we wanted to see if ants, but now we've come to realize that they exist in wasps, and even some my colleagues have told me that they found them in non-insect arthropods. um,
Wow.
so they are really, we are now calling them honeybee associated viruses because they're most commonly associated with honeybees, but it's not even fair to call them Hymenoptera which is the order in which bees are, that Hymenoptera viruses, because they can happen in non-Hymenopteran insects. So
Wow.
very interesting. The more we dig, the less we know about these viruses. But somehow at least some of these strains are mostly associated with honeybees, and they're the ones that are causing um, health issues.
So Miss Payne, right? She actually collected 77 different ant samples from across Texas.
Hmm.
57 were from apiary sites, 20 from non-apiary sites, and actually found some of the virus in a number of these ant samples, but didn't necessarily find the replicative genome. So what does
Yeah.
that mean? Why is that important?
So the specific ways that these viruses are detected molecularly are kind of difficult to explain. But
Mhm.
basically, there are several tests where you're looking for not just the genetic um, list of um, the genetic code of the virus to see if you can detect it within the ants, but you have to look at the the opposite sense of the strand that the virus would need to create if it is replicating within the organism to create more copies of itself. And
Gotcha.
so you have to have two um, two things going on for the replication to exist. The one is to find that the virus is within the organism,
Hmm.
um, but that you also find the replicative copy of kind of the opposite strand of the virus in order for it to replicate. And so you can find, which is what we found, you can find the genome, the RNA strand um, segments of the viruses within the ants, but not the replicative form, which
Yeah.
indicates that they're kind of just passively inside the ant, but it's not replicating within the ant. So we think that what's happening is the ants are eating bee um, products, so any sort of food, but mostly probably bee larvae and so not just honey and nectar, but anything that where the virus is, the
Yeah.
ants are consuming it, and they're just having it. They're more like mechanical vectors of the of the viruses,
Yeah.
uh, but not really replicating it within their bodies, which is, I guess, good. It's a good
Good thing.
side of
Yeah.
relief that they're not really replicating. I should say, Alex, and she would be better at explaining this. Alex did a series of uh, negative and positive controls in these
Hmm.
molecular tests that she learned to do along the way. And before she did the last very stringent types of controls, she had actually found that opposite sense strand of the virus in one or a few of her samples, indicating that they were actually replicating within the ant. And only after she did a couple more controls to test for false positives and false negatives did she realize that her that the results she had gotten previously were false positives for replication.
Wow.
And so we suspect that the couple of papers that have reported active replication of the viruses within the ants probably
They're
did not
false
run the
positives.
correct the controls.
Interesting. Yeah, because
Yeah,
in France, I mean, in France, at least there
yeah,
was a study that
yeah.
suggested that the carpenter ant had both the viral and the replicative genome. But I guess if they didn't have that particular extra test, they wouldn't know if they had a false positive.
That's right.
Oh,
And
wow.
I mean, that we'll just leave it there. But I think after so many testing so many bees and doing so many controls, we're very confident that the results we got
Pretty solid.
are solid, and that doesn't seem, at least from what it looked like in the methodology used by the other people, that they used as stringent of controls.
So as a mechanical vector, does that mean if an ant dies and its carcass is inside a beehive that it could somehow transmit that virus, or is it on the cuticle of the ant?
It's inside them, so they have
It's inside because
to.
they have to eat it.
Yeah, they eat them. So potentially you could see the virus getting transmitted if the bees come in contact with
The insides of the
the
ant
inside of
somehow.
the ant. But because bees are um, vegetarian, they're not going to likely doing that. But that brings a good point that that could happen with, let's say, a wasp that is carnivorous and goes and eats the ant. Then
Oh,
they could potentially get
that virus.
some of those variants inside, um, but who knows if they would replicate? Potentially not. But
Right.
still, they are reservoirs, let's say, of these viruses that at any moment could turn on the replication um, machinery inside the reservoir individual, and then start replicating.
Right. So if there is some kind of evolution or adaptation of that virus,
Right.
then that could cause an immediate switch and an immediate and
That's
a
right. I mean,
completely new
the
problem.
some of the work that has shown that these viruses occur in other non-apis bees, for like bumblebees, have shown the some of the viruses in flower parts, for
Wow.
example. And so when an infected bee comes and is consuming the food and potentially kind of regurgitating a little bit of it, and with some virus particles on the food, then another bee, say a bumblebee, can come to the same flower and then
And pick
get,
that up.
so they would need, face masks, tiny face masks, to avoid
Or sanitize your work surface
sanitize,
after you're
exactly,
done.
or the
Yeah.
tiny hand sanitizer dispenser to before eating, something like that.
Yeah. Well, Dr. Rangel, I want to thank you so much for taking your time today to talk to me about bees and your research, and really appreciate it.
Thank you so much, Irfan. I appreciate it. It was fun, and I'm really happy that we got to chat today.
I hope you've enjoyed this episode of Talking Bugs. Please leave a review or comments either on YouTube or your favorite podcasting platform, or join our Facebook group, Talking Bugs. Leave comments or share. That'd be greatly appreciated. Thank you so much. We'll see you next time.